Hi guys. Okay, so we have a biology paper here, Cambridge IGCSE, May June paper 4-2 for 2024. Alright, so we'll go through the paper and we'll discuss the questions that were asked in this particular paper. Alright, so let's go through it. Okay, so the first question here says, fungal cells and plant cells contain mitochondria. State the function of mitochondria. Alright, so mitochondria, this is one of the organelles that we find in a cell. Alright, so mitochondria is an organelle in which aerobic respiration takes place. Alright, and we know that respiration is a process that gives energy for different processes within the cell. But this energy will be made inside what? The mitochondria. So we'll say that the function for mitochondria is aerobic respiration. Alright, yes. Then state one feature of plants that is used to distinguish them from fungi. Alright, so the one feature that we can find in plants but not in fungi is the ability to photosynthesize. Alright, so because the plants can photosynthesize means they have chloroplasts. The fungus does not photosynthesize, so it means they do not have what? Chloroplasts. So one feature in plants that can distinguish them from fungus is what? Chloroplasts. It is the chloroplasts which contain chlorophyll and enables the process of what? Of photosynthesis. Alright, but in fungus we do not have chloroplast and does not photosynthesize. Alright, so we move further. Yeast is a fungus that can respire to produce ethanol. State the balanced chemical equation for this type of respiration in yeast. Alright, so yeast is respiring to produce ethanol, but this kind of respiration in which yeast is involved does not use oxygen. Okay, and the type of respiration that does not use oxygen is usually named as anaerobic respiration. Alright, so from here we need to state the equation for anaerobic respiration in yeast. Now, because oxygen is not involved in this process, it means glucose is breaking down by itself to give us ethanol and carbon dioxide. But how is it actually written? So glucose is usually written as C6, the chemical equation, H12O6. It's breaking down anaerobically to form what? Ethanol. So ethanol is usually 2C2H5OH plus 2 carbon dioxide. Alright, so this is the balanced chemical equation for respiration in yeast. Alright, yes, so that is how we write it. Okay. Then we go further. A scientist investigated the effect of sugar on respiration in yeast cells. One flask contained 100 centimeter cube of sugar solution, and another flask contained 100 centimeter cube of water. Both flasks contained the same mass of yeast. The temperature was maintained at 25 degrees Celsius. The scientists used the apparatus shown in the figure below, in figure 1.1. Alright, so they used this apparatus to investigate what? The respiration of sugar but in yeast. Okay, effect of respiration of sugar in yeast. Alright, so from the setup here we can see that we had solution of yeast and sugar in water. Alright, then we had an oil layer. And then this setup was also placed at a certain temperature inside the water bath, whereas the temperature was 25 degrees centigrade. We had a thermometer in there to read the temperature, and we had a syringe to collect the gas that is produced from the whole process of respiration. All right. Okay. Then the questions, the next graph here shows the results that were seen in the investigation. All right. So what is seen here is that as time went by, the volume of the gas that was collected, which is carbon dioxide, was increasing. That is what we can see. Right? Yes, so you can see as time increased from time 0 maybe to time 30, volume collected was 40, but as time went by, it also increased, and we can see that it became constant at a temperature of 30, at a time of 35 to 40 seconds. the temperature was, the volume of carbon dioxide collected was constant. All right, then the first question here says, using the gradient shown in figure 1.2, calculate the rate of carbon dioxide produced by the yeast solution between 10 minutes and 15 minutes. Include unit and show space for working. All right, so we have to read the volume of carbon dioxide produced at 10 minutes. All right, so 10 minutes is here. Okay, yeah, so at 10, volume produced was 10, 11. It was 11 centimeter cubed. So we will note here that at 10 seconds, volume of carbon dioxide produced was 11 centimeters cubed. All right, then we have to see the volume produced at 15 minutes. So 15 minutes is approximately here. All right, so we'll read the volume produced, whereas the volume will be at this point here. So the volume is going to be 19. All right, yeah. So at 15 seconds, we'll also note it down. At 15 seconds, the volume of carbon dioxide produced was 19 centimeter cubed. So we can see the time in between, the time frame is 15 minutes. All right, so we also need to find what? The rate. Now, to get the rate, we will take the volume obtained at 15 minutes and the volume obtained, we subtract the volume obtained at what? At 20 minutes. All right, so it will be 19 centimeter cubed minus 11 centimeter cubed. All right, when we subtract this one, we'll get 8. All right, so the difference that we get from the time given over here, the time at 10 minutes and the time at 15 minutes, will be 8. All right, but the time interval between the two between 15 minutes and 10 minutes is 5. All right, so in order to get the rate, we take the difference of the volume collected, we divide it to the time taken. All right, so it will be 8 divided to 5. Okay, then we work it out in a calculator. Let's do it. When you divide here, we'll get 1.6, but the difference here, the volume was in centimeter cubed and the time was in minutes, right? Yes, so when we divide here, it's equivalent to saying 1.6 centimeter cubed per minute. All right, yeah, so that will be the rate at which carbon dioxide gas is produced between 10 and 15 minutes. The answer will be 1.6 centimeter cubed per minute. Okay, then we move further. Suggest the reason for the oil layer in the apparatus shown in figure 1.1. All right, so why did they have to place a layer of oil up here in our apparatus? All right, yes, here. So they added the oil layer so that the yeast could respire anaerobically. And how so? It means there was no entry of oxygen gas in order to allow what? Anaerobic respiration. So the oil layer was there to prevent what? Entry of oxygen gas. All right, to prevent entry of oxygen gas. You prevent the oxygen gas from entering and dissolving into the water. All right, yeah, so that was it. Then state one reason why no more carbon dioxide gas was produced after 35 minutes by the yeast in solution, in sugar solution in figure 1.2. So here, after 35 minutes at this point here, the amount of carbon dioxide that was produced was constant. All right, so why so? Okay, so one of the things is that the substrate might have been used completely. Okay, so we do not have any sugar, we do not have any yeast maybe, so there's no more. Okay, there's no more production of what? Of carbon dioxide. So say the substrate has been used up. Okay, the substrate has been used up. All right, yes. So the substrate, the sugar, has been used up. Okay, yes. So if it has been used up, it means there will be no more production of carbon dioxide. Okay, the scientists repeated the investigation using yeast and the sugar solution at a temperature of 95. All right, so the temperature is so high. 95 is a very high temperature. All right, then explain why no more carbon dioxide was produced at a temperature of 95. So we need to tell why no more carbon dioxide was produced at a temperature of 95. All right, so the temperature is very high. Okay, so if the temperature is very high, it means there will be denaturing of the enzymes. The enzymes will be denatured. The enzyme active site will be denatured. So if the enzyme active site is denatured, it means there's no formation of enzyme substrate complexes. And if no formation of enzyme substrate complexes, it means there's no product that is formed. All right, so if no product that is formed, it means no carbon dioxide is formed at this point. Okay, so I would say here 95 degrees centigrade is a high temperature, that can denature the active site of enzymes. All right, yes. Then no substrate can bind to enzyme, hence no enzyme substrate formed, no enzyme substrate complexes are formed, and no products obtained. All right, so the temperature is very high to denature to the active site, and then at the end of the day, we will not have any product that is being formed. All right, then we go further. State one way in which humans use carbon dioxide gas produced by yeast. All right, so one way in which humans use carbon dioxide that is produced by yeast is in bread making. Okay, so the sugar will be mixed up in the flour, and at the end of the day we'll add yeast. Then yeast will respire anaerobically. As it is respiring anaerobically, it will produce carbon dioxide. This carbon dioxide will result into the rise of the dough of the bread. All right, and this is the essence of why humans add what? Use yeast in the whole process. They need the carbon dioxide so that the carbon dioxide can help in formation of the dough of the bread. All right, so would say one use of yeast for people, it's in bread making. Right, and then it says here, state the name of one other gas other than carbon dioxide that contributes to the enhanced greenhouse effect. All right, so the gases that can result into greenhouse gas effect other than carbon dioxide, one of them is methane. All right, so methane can be another gas that results into global warming or greenhouse effect. All right, then we move further to the next question. It says here, define sense organs. All right, so sense organs, these are organs that can detect changes in the environment. What changes are they detecting? The change that they are detecting is a stimulus. So a change in stimulus in a particular environment will actually be detected by the sense organ of an individual. Let's say light, for example. Light is a stimuli. So either increase or decrease in amount of light will be detected by the eye, as the eye is an example for sense organ. All right. Temperature will be detected by the skin, because the skin is an example of what? Of a sense organ. So say sense organs, these are organs that detect or respond to what? Changes in stimuli. Okay. The organs that detect. Right. Figure 2.1 shows the structure of the eye. It also shows the pathway taken by nerve impulse, which help bring about change in the eye in order to bring light into focus. Okay, so this is the diagram of the eye, and we can see it has different labeled path, and the pathway taken by nerve impulse as it is traveling from the part of the eye that allows light to get into until to the part of the eye that carries this impulse to the brain. Okay, then it says here, draw a label line and a letter X on figure 2.1 to indicate the motor neuron. Alright, so the motor neuron, this is a neuron that carries the interpretation of the impulse from the brain to the effector organ. Alright, it tells what the brain has interpreted about that stimulus, the brain or the spinal cord, and carries this interpretation to the effector organ so that the effector organ could act according to the interpretation from the central nervous system. Alright, while we also have another neuron known as sensory neuron. Sensory neuron, this one here, carries the impulse from the receptors to the central nervous system. And we have the relay neuron that is actually between the sensory neuron and the motor neuron. So it carries impulse between the two. Alright, so here, because you already know that the motor neuron is carrying interpretation from the central nervous system to the effector neuron, it means it will be pointing away from what? From the brain. Alright, and the neuron that is pointing away from the brain is actually this one here. Alright, this one here, because it is pointing away from the brain, so this would be the motor neuron. So we'll draw a label line to it and label it as X. And then describe how the events that occur at the synapse generate an impulse in the next neuron. Okay, so we have to understand that the impulse will come from the first neuron. We can call it presynaptic neuron. Alright, so they will come from the presynaptic neuron. Okay, when they reach at the region near the synapse, meaning at the edge of this neuron, they will meet some vesicles. These vesicles, in them, they contain the neurotransmitter molecules. Okay, so the neurotransmitter molecules will kind of bind with the impulse that has come along this neuron, and then the neurotransmitter molecules will diffuse with this impulse out of this presynaptic neuron into the synapse. Alright, so after they diffuse into the synapse, these neurotransmitter molecules will bind to the receptor molecules of the adjacent neuron or the postsynaptic neuron. After they bind to this neuron, what will happen? The impulse will get regenerated in this second neuron. We can actually demonstrate it here, that we have the first neuron, this one here, alright, and then we have the second neuron over here. This first neuron here, this one here, we can assume it's the presynaptic neuron because it's before the synapse. And then in here we have what? The vesicles. The vesicles contain neurotransmitters. These ones here can be the vesicles. But what do they have in them? They have neurotransmitters. So the impulse will bind with the neurotransmitters from the vesicles, then they'll diffuse into the synaptic gap here. And then after that, these neurotransmitters will bind to these receptors here in the second neuron. After they bind to the receptors in the second neuron, the impulse will be regenerated in this neuron, and it will keep on traveling across this neuron. That is how events are transmitted or impulse are transmitted to the next neuron. Alright, yes, so we'll say that neurotransmitters will be released from the vesicles and bind to the impulse. So neurotransmitters are released from the vesicles of the presynaptic neuron. Then they diffuse into the synaptic gap. Then we say that the neurotransmitters bind to the receptors of the postsynaptic neuron and the impulse is regenerated in the neuron. Yeah, so that is how events, these are the events that occur in case of impulse transmission. Okay, describe and explain the process of accommodation in the eye to view a near object. Okay, so the question wants us to explain how accommodation occurs to help us view a near object. Okay, so we have to know that, first of all, accommodation is the ability of the eye to view a distant or a near object. Alright, when viewing a distant object, the lens is made to become thin. Okay, how is it going to become thin? It means the suspensor ligaments over here, these ones here, the ones labeled K, what we call them the suspensor ligaments, and the ones labeled E are the ones that we call them the ciliary muscles. So when we are viewing a distant object, this part K, the suspensor ligament, usually contract or tighten. When they tighten, the lens becomes thin. Hence, the eye is able to view an object from a distance. But our question is talking about viewing near object. So when we are viewing a near object, it means the suspensor ligaments relax. Okay, so when they relax, it means the lens becomes, we can say it becomes fat and thick. Okay, it becomes big. Now, when it takes that shape, the lens is able to focus light from what? From a near object, and hence it can see what? A near, or hence the eye is able to accommodate a near object. So, what happens first is that the suspensor ligaments relax. Okay, suspensor ligaments which have been labeled, I have said here, they have been labeled as K. Suspensor ligaments K relax. That is the first thing. But as they relax, it means the ciliary muscles contract. The ciliary muscles contract. The ciliary muscles here have been labeled E. So ciliary muscles contract. Okay. Then the lens becomes, the lens loosen. The lens becomes loose, or we can say it becomes fat and short. Okay, yeah, so when the lens is short, then we can say then light from near object is focused on retina, and hence the eye is able to view a near object. Okay, the distribution of and function of rods in the eye. Describe the distribution and function of rods and cones in the eye. So we have to talk about how rods and cones are distributed and how they function inside the eye. All right. First of all, we need to know what is the function of a cone. Cone is a receptor cell in the eye, but also rods, they are also receptor cells in the eye. But cones, these ones here, are receptor cells that are sensitive to bright light, while rods are receptor cells that are sensitive to what? To dim light. Right? And then for that reason, cones usually form a bright and colored image, while rods form an image that is appearing like black and white or gray. Okay, that is it. So they are all receptor cells in the retina. So cones and rods are receptor cells in retina, receptor cells in retina. Okay, then we say cones are sensitive to bright light. They form colored images, and rods are sensitive to dim light. They form a black and white image. But then again, we have to talk about the distribution. Cones are usually found at the part of the retina called fovea. So cones are concentrated in fovea of retina. Okay, but rods are highly distributed in peripheral parts of the retina. Highly distributed in peripheral parts of the retina. Peripheral parts of the retina. But then again, we also have a part of the retina which is called blind spot. So we know that at the blind spot there is no rod or cone. Then we say that at the blind spot there is no rods and cones. So this is a part of the retina that doesn't have light sensitive cells. Alright, we go further. Red-green color blindness is a sex-linked characteristic. It is controlled by a gene on the X chromosome. There are two alternative versions of this gene. We have the one X, but it has a capital A as a superscript, and we have X then that has a small a as a superscript. Now the first question says, state the term used to describe an alternative version of a gene. Okay, so this thing or this term is usually known as allele. Okay, the allele of a gene is what is termed as the alternative form of a gene. And then state the genotype of a male with color blindness. Alright, so we have to know that for the sex-linked characters, the character is usually carried on the X chromosome, but not the Y chromosome. Alright, now because it is carried on the X chromosome, it means we don't expect the Y chromosome to be carrying this character. Okay, what will be carried in the X chromosome is what is going to be expressed. So for a male who has color blindness, it means the X is carrying the allele for color blindness, so it will definitely appear like that. Okay, yes. Yeah, then we move further. It says here, meiosis and mitosis are important processes in life cycles of organisms. Figure 3.1 shows the life cycles of two different organisms. Organism 1 has a simple life cycle. Organism 2 has a complex life cycle. It has stage A that produces spores and stage B that produces gametes. Now, some explanation on organism 2. Because it has stage A that produces spores, that means stage A is a form of asexual reproduction in this organism, and stage B produces gametes. So it means stage B is a form of sexual reproduction in this organism. Alright, in the diagrams, the haploid number of chromosomes is represented by letter N, and the diploid number of chromosomes is represented by 2N. So you have the first organism over here, as we can see. Alright, an adult has 2N, then process P takes place, which forms sperm and egg. Then we have process W taking place, which forms the zygote. Then we have embryo, then embryo we have an adult. That is the life cycle of organism 1. But for organism 2, we can see that it has a multi-stage A, which is diploid. Okay, then this diploid undergoes a certain division over here to form spores, which are now N. Alright, and then after N we have a multicellular stage B. Alright, multicellular stage B, which forms S, undergoes process S to form gamete and female, male and female gamete. Then we have zygote. Yes. Alright, now let's see what the question says. Okay, table 3.1 shows the letters P to T in figure 3.1 and the type of nuclear division. Place a tick in each row to indicate the type of nuclear division that occurs on each of the letters P to T. So we have two types of nuclear division. We have meiosis and we have mitosis. Meiosis, this one here, halves the number of chromosomes. Alright, it makes them haploid. So for example, if we had 2N, it means they'll be divided into N. While mitosis doubles the number of chromosomes. Alright, so if we had N, now we will have 2N. Alright, so let's see P first. Where is P? Alright, P here, it's a process in organism 1 which leads to formation of sperm and egg. All right, so sperm and egg, these ones here are usually haploid gametes, but they are coming from an adult which is diploid. So an adult which is diploid to form haploid gametes, it means this process taking place over here should be meiosis. So P will be meiosis. Okay, we need to see Q as well. Where's Q? Alright, so process Q is here. We have an embryo, but this embryo is going into something known as an adult. So in order for an embryo to become an adult, it means it has undergone multiple divisions which we can term them as mitosis. So process Q will be mitosis. Okay, then we move further to R, process R. Okay, so process R is over here, and we can see that it's from multicellular stage A, which is diploid, to spores, which are just having a haploid number. So again, process R will be meiosis. Move to process S. Okay, so process S, it's like we have multicellular stage of B, or haploid number of B, okay, which will then form N female gamete and what? And male gamete. So this stage here will also be mitosis. All right, then we have T, process T again. Where is T? So T again, we have a zygote growing into an embryo. That will also be mitosis. So we'll check mitosis as well. Okay, state the name of the process labeled W. W is here. Okay, it's a process in which sperm fuses with an egg to form zygote. So definitely this process will be fertilization. Alright, so we'll label it as fertilization. Okay, an embryo contains stem cells. Complete the sentences about stem cells and body cells. Stem cells are, we say stem cells are unspecialized cells. It means they're cells that have not specialized to do anything. So stem cells are unspecialized cells that divide. Okay, so they are unspecialized cells that divide by a process of mitosis. Okay, to form many more unspecialized cells, maybe unspecialized cells. So divide by mitosis to produce daughter cells that can become specialized. Okay, to produce daughter cells that can become specialized for a specific function. Most body cells in an organism contain the same genes, but many genes in a particular cell are not expressed. All right, multiple genes are found in an organism, but only certain genes are usually what? Expressed. So most of them we say they are not expressed, because the cell only makes specific proteins it needs. Okay, so you can see a difference in certain species of the same kind just because certain genes are being expressed while other genes are not being expressed. Okay, part B here says, state the events in life cycle diagram for organism 1 in figure 3.1 that would not be present in life cycle of asexual reproduction. Okay, organism 1, where is it? Which would not be present in asexual reproduction. All right, so organism 1, it definitely shows sexual reproduction. All right, so one of the processes that is present in sexual reproduction but not asexual reproduction is this one here, the process of fertilization. All right, so fertilization will not be present in these processes. Apart from fertilization, another thing we can speak of is meiosis, which is going to form these gametes. All right, so meiosis will not be there, fertilization will also not be there. So say meiosis, and then we'll say fertilization. Yeah, so these are the two processes that are not there. And then question 4. Some algae are single-celled organisms that can photosynthesize. Their cells contain a pigment chlorophyll. State the name of one mineral ion that is needed to make chlorophyll. All right, so we definitely have 2 mineral ions that are usually needed by plants. All right, most of the time we have magnesium, which is used to make chlorophyll, and we do have nitrates that are used to make proteins. All right, so here, because the question is asking about chlorophyll, so we will state magnesium as the answer. Magnesium ions. Okay, A student investigated the effect of light on photosynthesis in algae using hydrogen carbonate indicator solution. Carbon dioxide is an acidic gas, all right. Table 4.1 shows the color of hydrogen carbonate indicator solution at different pH values. We have it at a pH of 7.6, pH of 8.4, and pH of 9.2. And we also have color of indicator, whereas at pH of 7.6 it's yellow, at pH of 8.4 it's red, and at pH of 9.2 it's purple. So as pH increased, the color also changed. Then two test tubes were set up at the same time. Both contained algae and hydrogen carbonate indicator. At the start of the investigation, the hydrogen carbonate indicator was red in both test tubes. One test tube was placed in the dark and one test tube was placed in the light. After 20 minutes, the contents of the test tube in the dark were yellow and the contents of the test tube in light were purple. Explain these results. All right. So after some time, we can see that the contents in one test tube were purple while the other one were yellow. All right. So for the one that were purple, it means that the pH increased. Okay. So we can say that after 20 minutes, the contents in test tube in the dark were yellow and the contents in test tube in light were purple. So in purple, it means pH increased. All right, that's why they turned purple. But the one that were in yellow, it means the pH did not increase. But then again, because we used hydrogen carbonate as an indicator, for the contents in light to turn purple, it means photosynthesis is taking place for the test tube that is in purple, is in the light. All right, so photosynthesis is taking place. Carbon dioxide is being used. Now, you know carbon dioxide is an acidic gas. It was even stated over here. So carbon dioxide is being used, hence the pH is doing what? Is increasing. All right, acidity is decreasing, pH is increasing, and hence the contents are turning purple. But for the one in the darkness, it means the rate of photosynthesis is low or not taking place at all. Okay, so if photosynthesis is not taking place at all, it means carbon dioxide is not being used. Instead, more carbon dioxide is being produced from the process of respiration. Hence, the contents are yellow. So I would explain the results and say that the test tube in light is undergoing photosynthesis. The carbon dioxide produced is absorbed for the photosynthesis process. But also the content of carbon dioxide decreases, hence the pH increases, and the contents turn purple. All right, then the test tube in light, the test tube in dark is not photosynthesizing. All right, carbon dioxide is produced from respiration, hence increase in its concentration, and the contents turn yellow. So this is what is observed from this setup. All right, we move further. Figure 4.1 is a graph showing effect of light intensity on rate of photosynthesis. As we can see here, on the y-axis we have the rate of photosynthesis, on the y-axis we have light intensity. Okay, so state which factors could be limiting the rate of photosynthesis at x and y. Alright, so as you move further on your y-axis, the amount of light is what? Is increasing. Alright, but when we have reached maximum amount of light, which means light is not at this point here, which means light is not affecting the rate of photosynthesis anymore. Alright, so at this point here, other factors will be affecting the rate of what? Of photosynthesis. So at point X, when light was increasing, it means that light was the what? The factor affecting rate of photosynthesis. So we would say that at X, the factor affecting rate of photosynthesis was light intensity. But when it came to point Y, it means light is not affecting rate of photosynthesis anymore, so it is something else. So at Y, we can speak of carbon dioxide. Carbon dioxide, concentration of carbon dioxide could be a factor that is affecting photosynthesis. We could even speak of temperature also. Yeah, so these are factors that can affect photosynthesis at this point. Okay, then starch and sucrose are made by plants after photosynthesis. State the name of the tissue that transports sucrose around the plant and the name of one other biological molecule that is transported in this structure. Alright, so we have two tissues in plants that are doing the role of transportation. We do have xylem tissues, which are specialized in transportation of water and mineral salts. But we also have the phloem tissues, which have specialized in the role of transporting what? Assimilates, such as glucose. Right, so the name of the tissue that is going to transport these substances or these assimilates is the phloem. Alright, now one other substance they can transport apart from sucrose is amino acids. Alright, yeah. And then describe how starch is broken down in the human digestive system so that it can be absorbed in the blood. Alright, so the digestion of starch usually starts in the mouth. Okay, in the mouth, starch is broken down by the process of physical digestion, which is an action of teeth. Alright, but also in the mouth there's an enzyme known as salivary amylase. Okay, so salivary amylase usually acts on the starch and it can result into formation of maltose. Then this breakdown of starch will then be carried through the esophagus into the stomach, where churning will take place. Okay, and then you'll have hydrochloric acid that will kill any bacteria eaten with food. Then this food will move from the stomach into the small intestine. In the small intestine, there is amylase, which is produced from what? From the pancreas. Right? Yes. So this amylase will again act on the starch to break it down into glucose, but also will have maltose in the small intestine. Maltose will break down this maltose, which was formed from incomplete breakdown of starch. It will break it down into glucose. Now glucose, this is the smallest unit in which starch can be absorbed in the blood, and this absorption will also take place within the small intestine. Alright, so we would say here starch is broken down, the digestion of starch in the mouth by action of teeth to increase surface area for enzymes to act. Then salivary amylase breaks starch into maltose. Okay, then in the stomach the starch or the mixture is churned, mixing. And then again we can say that hydrochloric acid also kills any pathogen within it. Then in the small intestine, amylase, pancreatic amylase, breaks starch into maltose, and then maltase breaks maltose into glucose. All right, then we say the glucose is then absorbed from the villi of the small intestine into the blood. Yeah, so this is what is happening during the process breakdown of starch in the body. Okay, then we have question 5 over here. Question 5 says 5.1 is a photograph of a koala. Koalas are marsupial mammals that give birth to offspring that are incompletely developed. The offspring develops further for several months inside the pouch of the mother. State one other visible feature in figure 5.1 that could be used to identify koala as a mammal. Alright, so one of the features that we see in mammals, first of all, is fur or hair. So when you look at the koala over here, you will see that its body is covered with what? With fur. But also we can see ears. You can see ears over here. It has large external ears, but also the body is covered with what? With fur. So we can say that it has what? Hair, fur or hair, which we see, but also you can speak of what? Of large external ears. Okay, mammals use sexual reproduction to produce offsprings. Discuss the advantages of sexual reproduction in organisms such as koala. So one of the greatest advantages of sexual reproduction is that it creates genetic diversity. Okay, genetic diversity, this one here helps these species to be able to survive in different conditions. They can adjust and be able to adapt to these different environmental conditions. All right, yes, so it allows for genetic diversity, but also another thing is that it reduces chances of extinction. And most especially when these organisms have been exposed to certain harsh environments harsh conditions in the environment, you find that they are able to resist because of their difference in genetic makeup. Okay, but also we can say that it allows for adaptation. Why adaptation? Because they have different in genetic makeup, but also they can be resistant to what? To unpredictable environment conditions such as what? Diseases. Yeah, so those are some advantages of sexual reproduction in organisms such as koala. All right, then here we have a diagram of a part of the human female reproductive system viewed from the side of the body. Then complete table 5.1 by writing the letters that identify the structures in figure 5.2, the names of the structures and their function. All right, so we have the name here, which is the cervix. We have to identify the cervix and its function. So the cervix, this is the part that dilates to allow childbirth. Okay, so that is the function. We can say that it dilates to allow childbirth. This is what it does. But then again, we have to identify the part which can be labeled as the cervix. So the cervix is just here, right? Yeah, so it is going to be part M. Okay, then ovary. So the ovary, this one's here, usually release mature eggs. Okay, so they release eggs and we can actually point them out here. So here, this one here is going to be the ovary. All right, so we'll say that it releases eggs, or you can say release eggs. Okay, and we have already identified that it was part L. Then we have site for fertilization. So fertilization usually occurs within the fallopian tube, which is this part here. All right, yes, so we'd say site for fertilization is J, and it is usually called fallopian tube. Okay, then site for implantation. So implantation usually occurs in the uterus. Okay, all right, so here it means that we have to identify the uterus, and the uterus is going to be this part over here. This is the uterine wall. All right, so it is labeled P. Okay, so site for implantation is P, and the name is uterus. All right. Then pregnant human females have a placenta. Describe the function of placenta in humans. So a placenta, this is a part or an organ that allows for exchange of substances between a mother and a child. All right, so we will name this exchange, how it facilitates this exchange. First of all, the function is, we say, to allow exchange of gases, which means to allow exchange of gases between the mother and the growing fetus. All right, yes, that's one thing. Another thing is that it transports nutrients from mother to fetus. It allows exchange of nutrients from the mother to the fetus. All right, but also we can say that it allows transportation of what? Of wastes. So we can name it as transfer of wastes from fetus to the mother. From fetus to the mother, so that they can do what? They can be excreted. All right, yes, but also it transfers, or it allows, or it produces hormones. All right, so the placenta usually produces hormones that are needed by the child, but also it usually transfers antibodies, which are very necessary to the growing fetus for the purpose of what? Of protection. All right, yes, so those are the functions of the placenta in humans. All right, the placenta is connected to the amniotic sac. State two functions of the amniotic fluid that is found in the amniotic sac. So here we have to state the functions of amniotic fluid. All right, so one of the functions of the amniotic fluid is that it nourishes the fetus. Okay, it prevents it from drying, so it keeps on nourishing it. But also we can say that it allows movements of the fetus. Right, you see some pregnant women, they can say the baby is kicking, the baby is moving, because the amniotic fluid is in there to allow movement of the fetus. All right, yes. Then question 6 here says, complete the sentences about human teeth. The process of taking food into the mouth is called, the process of taking food into the mouth is usually called ingestion. All right, yeah. And then flat, sharp edged teeth at the front of the mouth are called, so the sharp teeth at the front of the mouth, which are used for biting of pieces of food, these are the incisors. All right, yeah, so we'll name them as such, incisors. All right, then they are used for biting of pieces of food. The dash and dash are large teeth. Okay, so these large teeth here are the premolars and molars. These are the large teeth. Okay, yes, towards the back of the mouth, and they are usually used for chewing. You use them for chewing, for crushing, or for grinding food. Right? Yeah. Then the next question says, figure 6.1 in the diagram of a villus. All right, yeah, as we can see it over here. Then draw labels and label line on the figure 6.1 to identify capillary and lactea. All right, so lactea is usually this central part here, this one here, this central part over here. So this one here will be the lactea. All right, yeah, and then we have to name the capillaries also. A capillary. All right, so capillary is a network of blood vessels within the lactea, and this is going to be a capillary. Okay, then the next question says, state the part of the digestive system where villi are located. So villi are usually located in the small intestine. In the small intestine is where we find villi. Okay, then the next question is state the function of a lactose. So as we have seen the lactose over here, it usually has a function. What is its function? Its function is to absorb and transport fats. So it absorbs or transports fats. That is what it does. And then describe the pathway taken by the products of protein digestion from the villi to the liver. So after they have been absorbed in the villi, the first thing, they will go into what? Into the capillaries. All right. Then after they get into the capillaries, they will get into the hepatic portal vein. That is a blood vessel that will carry them from what? From the villi into the liver. All right. So the contents will do what? Will move from the villi into the capillaries. All right. Okay. So amino acids into the capillaries of the villi. All right. Then into blood plasma. Okay, into blood plasma, into the blood inside the capillary. Then to the hepatic portal vein. To the hepatic portal vein that transports them to the liver. That transports them to the liver. All right, yeah. Okay, and that was the last question for this paper, May June 4 to 2024. Thank you.